up, biology students? Your benevolent biology teacher here. In today's video, we're going to talk about how the theory of evolution by natural selection has influenced our approach to classifying living organisms, and two related but slightly different methods for doing so, disciplines called cladistics and phylogenetics. In our previous video, we talked a little about Linnaean classification, a system for naming and classifying living organisms invented by Swedish scientist Carolus Linnaeus in 1735. This system predates the publication of Darwin's book On the Origin of Species by over 100 years, but in many ways it's pretty amazing how well it has lasted despite a huge paradigm shift as to where species come from. According to this system, the largest, broadest category of organisms is the kingdom. Plants belong in one kingdom, animals in another, fungi in another. At one time we recognized 5 kingdoms, which included Protista and Monera as kingdoms as well. Protista included the single-celled eukaryotic organisms, while Monera included the single-celled prokaryotic organisms. Within each kingdom there are phyla. Within each phylum there are classes. Within each class there are orders. Within each order there are families. Within each family there are genera. And within each genus there are species. And a species is a specific variety of organism. As we've learned more about biology over time, especially in regards to evolution and genetics, we've revised our system for classifying living organisms. The current system includes 3 domains as the broadest categories of life: bacteria, archaea, and eukarya. The domain bacteria contains, you guessed it, all the bacteria, single-celled prokaryotes that lack membrane-bound organelles. The domain archaea also contains single-celled prokaryotes, but there are some fundamental differences between archaea and bacteria, having to do with the cell wall and membrane structure, niches inhabited, and key genetic differences. The archaea are often referred to as extremophiles because they inhabit environments that are too extreme for other life forms. Hot springs, extremely salty environments, and environments that lack oxygen and contain methane gas instead, for example. The domain eukarya contains all the organisms, both single-celled and multicellular, that have eukaryotic cells, larger cells with membrane-bound organelles. From an evolutionary perspective, the separation between domains is the first huge evolutionary distinction between organisms. All eukaryotes, from flowers to fish to fungi, share an evolutionary heritage and share key similarities in cell structure. They inherited these from a very, very ancient population of single-celled organisms that had these characteristic cellular structures. And remember, cells are the basic unit of life. So if this group has that in common, they must be pretty similar in an evolutionary sense. In fact, the archaea and the eukarya also share a common ancestor, one that is even older than the common ancestor of all eukaryotes. And both archaea and eukarya share a common ancestor with a bacteria that is even older, roughly 3 to 3.5 billion years old, according to the fossil record. Where those original cells came from is still something of a mystery, but mysteries are what drives and motivates science. So, of course, there are hypotheses, and many people working on trying to find a scientific answer to this question. And, of course, there are other perspectives that are rooted in philosophy, spirituality, and personal beliefs, but these are all stories for another day. Suffice it to say that wherever those first cells came from, evolutionary processes have been at work since the beginning of their existence, shaping the life on this planet and producing the biodiversity that we see around us today. And these days, we classify this biodiversity according to evolutionary history. The practice of cladistics is one method of classifying organisms based on their evolutionary relationships, and we assess and determine these relationships using the similarities in morphology, or physical appearance, and the presence of derived traits, which we'll talk more about in a minute. This figure is called a cladogram, and it is a model that depicts the evolutionary relationships between organisms based on their physical characteristics. Specifically, cladograms show how evolutionary lines, or lineages, split from each other over time due to speciation. The first cladogram shows one lineage splitting into 2. This spot on the cladogram, the point where 2 lineages diverge, is called a node, and it represents the common ancestor of all lines diverging from this point. In this second figure, we see a slightly more complicated cladogram. For example, we can see that this line has 3 representatives. Each number represents a taxon, or group of organisms. Could be a species, or a genus, or a family, or something as broad as plants or vertebrates. The group size doesn't matter, as long as that taxon, or number in this case, represents all the organisms that evolved from this evolutionary line. In this case, we have a cladogram with 4 taxa. Taxa 3 and 4 are the most closely related because they share the most recent common ancestor, represented by this node here. It is important not to confuse closeness on the cladogram with how closely related 2 groups are. It would be a mistake to say that the closest relative of taxon 1 is taxon 2 because taxon 1 is equally related to all 3 of the others. Think of taxon 2, 3, and 4 as all being part of the same group, the same evolutionary line. Taxon 1 is equally related to that whole group because they all share the same common ancestor way down here at this node, which means this common ancestor predates all 4 lineages shown here. This last cladogram is even more informative. It shows the relationship between 9 representatives of the vertebrates, all the animals with vertebrae protecting their spinal cord. This cladogram obviously does not contain every single species of vertebrate, but it does show us most of the major groups, and each of these groups, or taxa, could be expanded to show the evolutionary relationships between more specific groups within that taxa. For example, we could have a cladogram to show us the relationship among all the mammals, or all the amphibians, or all the sharks. That's kind of the cool thing about cladograms. We can view them at any scale, zoomed way in or zoomed way out. This one is zoomed pretty way out, but it does show us how the major groups of vertebrates are related, as well as how recently these groups share a common ancestor with one another. For example, we can see that the lizards and snakes share a much more recent common ancestor than the lizards and turtles, which means that the lizards are much more closely related to snakes than they are to turtles, even though we would classify them all as reptiles. A clade is any group that contains all the descendants of a single common ancestor, and clades can be any size as long as they meet this criteria. For example, this cladogram shows 5 different clades. The first and largest clade is the Tetrapoda, which includes all the taxon on this cladogram. This text here describes a derived characteristic, and a derived characteristic is an important trait that evolved in the common ancestor of a particular group, and which defines that group. It's important to note that you can't just go around picking any random physical trait to use to build a cladogram. It matters which traits you choose, and you have to choose traits that are evolutionarily meaningful. Having 4 limbs is a derived characteristic on the clade of Tetrapoda because all the members of this clade have 4 limbs, a trait that they inherited from their shared common ancestor. And having 4 limbs is an evolutionarily really important trait. That's a big modification or a big change from a body plan that doesn't have 4 limbs. Having brown eyes or blue eyes, however, is not an evolutionarily meaningful trait. The next largest clade is the clade Amniota, which includes all the organisms on this cladogram except the amphibians. All the taxa within this clade have an amniotic egg, an egg surrounded by an extra protective membrane. And this is a derived characteristic for this clade because it evolved in the common ancestor of all of these organisms and was passed on throughout each of these evolutionary lineages to follow. The amniotic egg is an important trait because it allowed organisms to move away from the water and lay their eggs on dry land, and that amniotic sac prevented these eggs from drying out in a way that, for example, a frog's eggs would if it tried to lay it on dry land. The next largest clade is the clade Mammalia, the mammals. This clade contains all the descendants of this common ancestor, which means it does not contain the amphibians or reptiles. Hair is a derived characteristic of this group, and all members of this group inherited their hair from their shared common ancestor. As you can see, these clades are becoming increasingly specific as we go up the tree, because each clade contains fewer groups descended from a more recent common ancestor than the clade before it. The next clade is the Carnivora, all the mammals with specialized shearing teeth for a diet comprised primarily of other animals. This clade contains the dogs and the cats, but does not contain the amphibians, the reptiles, or the marsupial mammals. The last clade on this cladogram is the clade Felidae, the cats, including house cats, tigers, lions, pumas, leopards, jaguars, and more. Retractable claws is a derived characteristic of this group, having evolved in the common ancestor of all cats, and being passed on to every lineage within this clade. Common ancestry is a huge part of modern classification systems, and helps us to see some of the problems with the traditional Linnaean classification system. For example, according to the Linnaean classification system, the class Reptilia contains the turtles, lizards, snakes, and crocodiles, as well as several other major groups, but not the birds, because birds are physically and behaviorally pretty different from, say, lizards. However, this is not an evolutionarily appropriate group because it is not a clade. To be an acceptable clade, a group must contain all the descendants of a particular common ancestor, which means that the clade Reptilia must include the birds because birds share a common ancestor with the lizards, snakes, turtles, and crocodiles. That's why they have scales on their leg. They inherited these scales from their shared common ancestor with the rest of the reptiles. Birds on their own are also a clade because all birds share a single common ancestor, but without the birds, the group that we might call the reptiles is not an evolutionarily meaningful group. And that's the problem with the Linnaean classification system. The clade Reptilia also contains the dinosaurs, which are actually more closely related to birds than they are to the lizards, crocodiles, or turtles, because birds and dinosaurs share a more recent common ancestor. And birds share an even more recent common ancestor with dinosaurs like the Velociraptor and T-Rex. From an evolutionary perspective, birds are dinosaurs because they are in the same clade. Just like the group reptiles must include the birds because birds share a common ancestor with the rest of the reptiles, the group dinosaurs must also contain the birds because they all share an even more recent common ancestor. That's pretty cool because it means that not all the dinosaurs went extinct. Some of them are still around, alive and well, eating in our bird feeders, perching on telephone poles, and picking up worms after it rains. In this table, we see a list of 7 different groups of organisms, as well as marks to indicate whether or not these groups possess certain derived traits. These derived traits include having a dorsal nerve cord and notochord, which is basically a flexible rod like a spine that runs down the back parallel to the dorsal nerve cord, having bony vertebrae and a jawbone, having 4 limbs, having amniotic eggs, having mammary glands and hair or fur, having a placenta, and having short canine teeth and a foramen magnum that is more forward in the skull. The foramen magnum, by the way, is the hole through which the spinal cord exits the skull. In most tetrapods, this hole is on the back of the skull because most tetrapods walk on 4 legs and have a spine that runs parallel to the ground. So it makes sense that they would evolve to have a spinal cord that comes straight out the back of their skulls. Even monkeys have a foramen magnum that is further towards the back of the skull because they can stand on 2 legs but rarely walk around that way. Humans are the only group of organisms in this table to possess this derived trait. In fact, humans possess all these derived traits. So let's start our cladogram with the human branch, and let's label this branch with the derived characteristic that defines this group and no other, the short canine teeth and the forward foramen magnum. The group in this table that has the most in common with humans are the rhesus monkeys. In fact, they share all the traits with humans except for the short canines and forward foramen magnum. Moreover, the rhesus monkey also has a placenta, which is a trait that no other group on this table has except for the humans. So let's put the rhesus monkey on the cladogram next, and let's label the group that contains both rhesus monkeys and humans with the derived trait that defines this group, having a placenta. One group in this table shares all these traits with the humans and monkeys except for the 2 traits that we've already put on the tree, and that group is the kangaroos. Kangaroos also share a trait with rhesus monkeys and humans that is shared by no other group. All 3, humans, kangaroos, and rhesus monkeys, have mammary glands or hair or fur. So let's add the kangaroo to our cladogram next, and let's label the group that contains humans, rhesus monkeys, and kangaroos with the derived trait that defines all 3, mammary glands and hair or fur. Hopefully by now, you're starting to see a pattern. Next on this cladogram, we should put the snapping turtle, because snapping turtles share a trait with humans, rhesus monkeys, and kangaroos that is possessed by no other group listed on our table. All 4 of these groups have an amniotic egg, which is the derived trait that defines the group that contains humans, monkeys, kangaroos, and turtles. Next, we're going to place the bullfrogs, because they, like humans, monkeys, kangaroos, and turtles, are tetrapods, meaning they have 4 limbs, while the other groups listed on our table are not. Next, we have the tuna, which has bony vertebrae and a jawbone just like the human, monkey, kangaroo, turtle, and frog, whereas the final group listed in our table does not have this trait. That final group is the lamprey, a chordate who lacks a bony spine and which doesn't have a jaw. It does, however, have a dorsal nerve cord and a notochord, traits shared by all the organisms in our cladogram. This trait evolved in the common ancestor of all chordates and was inherited by all the organisms to descend from this ancestor. It is also important to note that we only have a few of the many important derived traits that are present in members of this clade. For example, scales evolved here, in the common ancestor of fish and all the other groups at this point on the tree. However, scales were lost in the amphibians and evolved to become hair or fur in the mammals, these 3 groups at this end of the cladogram. Likewise, claws evolved here, in the common ancestor of turtles, kangaroos, monkeys, and humans. And these claws evolved to become fingernails in the group that contains monkeys and humans. In our example, we built our tree from the top down, from the organisms that share the most recent common ancestor to those that share a more distant common ancestor. That's pretty much the opposite of the direction that evolution goes, and we sort of went backwards through evolutionary history. Another way to build our tree would be to start at the base and build our way up, which is more like how evolutionary history actually played out. So if we did it that way, we'd start with a lamprey, which only contains 1 derived trait, the first one to evolve in the evolutionary history of this entire group, and one shared by the lamprey and every other group on our list, the dorsal nerve cord and a notochord. Next, we'll add the tuna, which also has a dorsal nerve cord and notochord, but which also has bony vertebrae and a jawbone, traits the lamprey does not possess because it diverged before this trait evolved. Next, we'd add the frog, which has a dorsal nerve cord and notochord, bony vertebrae and a jawbone, and also 4 limbs, a new trait that neither the tuna or the lamprey possess because they diverged before that trait evolved. Next, we will add the turtle, which has all the traits we've listed so far, but which also has an amniotic egg, a new trait that the frogs, tuna, and lamprey did not have. Next, we'll add the kangaroo, which has all of the traits below, but which also has hair or fur and mammary glands. Then we'll add the monkey, and finally the human, both of whom have a placenta. The human also has short canine teeth and a forward foramen magnum. Building our tree this way showed us the order in which certain traits evolved in the history of life on Earth, from the oldest traits, like the dorsal nerve cord, to newer traits, like the placenta in humans and monkeys. And if you're at all curious as to what this common ancestor might have been like, well, we think we found something pretty similar in the fossil record, an organism called Pikaia that lived approximately 530 million years ago. Fossil evidence suggests that Pikaia had a notochord, and likely had a dorsal nerve cord as well. It was a marine organism, pretty small, and without any hard bony features, but with all the defining characteristics of chordates. From a population of organisms very similar to this, all the animals with a spinal cord have evolved. So cladistics is a pretty good system for classifying living organisms according to their evolutionary relationships, but even this practice is somewhat out of date. An even more modern approach involves analyzing genes and genetic similarities in order to determine evolutionary relationships, because genes don't fool us the way that morphology sometimes does. This practice is called phylogenetics, and it is very similar to cladistics in most important ways, except that it is genes and not morphology that determines how we put our tree together. We call these trees phylogenies, or phylogenetic trees, instead of cladograms. For example, here we see a phylogeny of animals based on specific Hox genes possessed by these representative animals. As we can see, organisms that share more recent common ancestors have Hox genes that are overall very similar. These 2 groups of organisms here, for example, have nearly identical Hox genes, and they probably share a very recent common ancestor. These 2 groups, however, have pretty different sets of Hox genes, and are likely to have a much more distant common ancestor. We can also compare intron sequences between different groups. Mutations to these intron sequences do not affect the organism because these introns are not expressed and are not being actively used for building proteins. So these neutral mutations tend to stick around in a lineage because they do not negatively affect fitness. These kinds of neutral mutations are the ticks of the molecular clock. The more similar the intron sequences of two groups, the more closely related they are. And the more different the intron sequences, the more distantly related the two groups probably are. That's because these mutations happen at a pretty constant rate, and the more time you allow to pass, the more mutations are likely to occur. For example, in this graph, each point represents a comparison of an intron sequence for two specific groups of animals. We have time on the x-axis and differences in nucleotides on the y-axis. So, the further to the left a point is on the graph, the more recently two groups of organisms diverged, and the more closely related they are. The further to the right a point is, the more distant the common ancestor of the organisms listed. The lower down a point is on the graph, the fewer differences there are in nucleotides, and the higher up a point is, the more differences there are. As you can see, there is a very clear correlation. As time increases since divergence, so does the number of nucleotide differences, which is to say that the further back in time two groups share a common ancestor, the more different their intron sequences will be because they've had more time to accumulate mutations. This kind of data, and the pattern depicted in it, helps us to make predictions about when two groups of organisms shared a common ancestor. Humans and kangaroos share a common ancestor that lived approximately 125 million years ago, according to this scale, while horses and cows share a common ancestor that lived about 75 million years ago, and sheep and goats share a common ancestor that lived only about 10 million years ago. Since proteins are coded for in our genes, and since DNA instructs our cells how to put amino acids together to build these proteins, we can analyze the genetic similarities between two groups of organisms by analyzing the sequences of amino acids present in proteins manufactured by the cells of those organisms. The more similar those amino acid sequences, the more closely related the groups must be, because the more similar their DNA must be. Here we see amino acid sequence differences listed for several different species. Specifically, we're looking at a protein called hemoglobin, or at least a piece of it, and how it differs in its amino acid composition in these 6 organisms. Humans and gorillas have hemoglobin that differs by only 1 amino acid, whereas humans and rhesus monkeys differ by 8 amino acids, and humans and mice differ by 27 amino acids. This tells us that humans and gorillas are the most closely related group on this table due to the similarities in their hemoglobin structure, which is of course determined by the genetic sequences of each group. It is precisely this kind of data that has helped us to find our place among the animals in the phylogenetic tree of life. For whatever protein we are looking at in this dataset, and honestly I'm not quite sure which one it is, humans, gorillas, and chimps have identical amino acid sequences, 0 differences. Baboons, who also make this protein, make it slightly differently, and there are 5 amino acids that are different when comparing the protein found in baboons with the protein found in humans. This same protein contains 7 amino acid differences when comparing humans and lemurs, telling us that humans are far more closely related to chimps and gorillas than they are to lemurs and baboons. And remember, it's genetics and DNA that tell us this is so, because genes ultimately code for these amino acid sequences that we see in this table. While we're on the subject of humans, it's time to dispel a particularly persistent misconception about human evolution. The idea that somehow humans evolved from chimps, or from monkeys, or from any other modern primate for that example. This is simply not the case, but it is something that people say from time to time. If humans evolved from chimps, then why are chimps still around? Well, the answer to that challenge is that no one who understands evolution would ever claim that humans evolved from chimps. That's not what we're trying to say in this evolutionary tree. Humans and chimps are contemporary species, modern species that live at the same time in history. One didn't evolve from the other any more than you descended from your cousin. Likewise, humans didn't evolve from chimps, we merely share a common ancestor with the chimps, one that lived millions of years ago. But humans and their ancestors, as well as chimps and their ancestors, have each been evolving along their own separate evolutionary lines since that divergence occurred 5 to 10 million years ago. And the common ancestor of humans and monkeys lived even farther back in time. This image, which is pretty iconic and recognizable, misrepresents the theory of evolution because it suggests a line of descent that goes like this, with something like a monkey evolving into something like a chimp, which is evolving into something like an upright proto-human, and so forth. But that's just not how it works. A more accurate picture of evolution would be something like this: an evolutionary tree that shows the common ancestry shared by these representatives of the human family tree. So which is better for determining evolutionary relationships, cladistics or phylogenetics? Well, the scientific community commonly accepts that genes are more reliable than morphology for several reasons, not the least of which is the fact that convergent evolution has a way of producing organisms that look similar, and which may even behave similarly and occupy similar niches, but which are quite distantly related. Convergent evolution is when distantly related lineages evolve into similar forms because of similar environmental and selective pressures. Here in this diagram, we see a bunch of examples of convergent evolution between marsupials and placental mammals. Marsupials, called here the Australian mammals, are mammals because they have hair and mammary glands, but they are quite different than placental mammals like us. In marsupials, offspring are born very immature, even compared to how our offspring are born. They're pretty much still fetuses. And they're raised in an external pouch where the mother provides them with milk and protects them from the elements. In placental mammals, the fetus remains inside the mother for a great deal longer, which is made possible by the placenta, and the offspring is able to continue to develop in the womb rather than being born and raised in an external pouch until fetal development is complete. But in many ways, evolution has produced animals with very similar forms, but which belong to very different groups. And even if they look similar superficially, genetic analysis would reveal vast differences between the placental mammals in this column and the marsupial mammals in this column. In fact, every animal in this column is more closely related to every other animal in this column than it is to any of the animals in the other column. Another interesting example of convergent evolution is the case of sharks, dolphins, and ichthyosaurs. All three live in the ocean and occupy similar niches and have similar morphology as a result. But they belong to very different evolutionary groups. The shark is a fish and has been evolving along with the fish for millions of years. The ichthyosaur is an ocean-dwelling dinosaur, one that evolved from ancient reptiles living on land. It is most definitely not a fish, even if it looks like one. And if it was alive today, genetic analysis would reveal this difference even more strongly than the fossils left behind do. And finally, dolphins and porpoises are mammals, not fish and not reptiles. These are fairly obvious examples, and we could probably figure out that sharks and dolphins don't belong in the same group, even without the genetic evidence. But the point is that morphology can fool us in a way that genes cannot. And it isn't always easy to distinguish groups without genetic evidence to help us sort out and identify the real differences. For example, the tree on the left, tree A, is a cladogram of the mammals based on morphological evidence, and the tree on the right, tree B, is a phylogeny of the exact same mammals based on genetic evidence. Individual groups have the same color in both trees to make it easy to see the differences between these trees. As you can see, some relationships remain the same, but there have been several places where groups got moved around a lot after genetic evidence became available, and the color coding helps us to see this reshuffling. Derived traits are still important, but using genetic evidence to build the tree, without allowing ourselves to be fooled by superficial similarities in form, and then looking at the derived traits possessed by different groups on different parts of the tree, helps us to better understand the evolutionary history that produced this diverse group of organisms. Now that you know how evolutionary trees are put together, and how morphological and genetic evidence are used to help us determine evolutionary relationships, I encourage you to slow this part of the video down and take a good long look at this evolutionary tree scrolling behind me. Look for how different groups of organisms are related to one another, and where different groups branched off from one another along their own independent evolutionary trajectories. See if you can find the evolutionary lineage that leads to plants and the one that leads to fungi. See if you can find the mammals and the reptiles, including the birds. See if you can find some of your favorite creatures on the tree, and then see if you can tell which other organisms they are most closely related to. Because that's the beauty of evolution. It helps us to understand where all of this magnificent and wonderful biodiversity came from, and how the different groups of organisms that make up our planet are related to one another. It helps us to understand our own origins and how we fit into the animal kingdom, while also helping us to understand how we are unique and special in our own ways. It helps us to see the order in the apparent chaos that is life, and that's pretty amazing if you ask me. And with that, I will bring our video to a close. Thanks for watching, and remember, you can go back and watch this video as many times as you need to until you feel like you understand the practices of cladistics and phylogenetics and the role of evolution in our modern biological classification systems.